During this mini lecture, we are going to be talking about meiosis and how we produce sperm through the process of meiosis. What I don't want to do is spend an hour reviewing what meiosis is in that process. That is something that you should have gotten in previous classes in BIO100 or 101 or genetics or something. So if you don't remember what meiosis is, I would strongly encourage you to actually pause this for a minute or turn it off and go find a review video. There are all sorts of review videos. There's Khan Academy videos, there's crash course videos, there's amoeba sister videos, whatever works for you, but review the concept of meiosis, because this is not going to make a lot of sense unless you understand how meiosis works and sort of what the end product or goal of meiosis is. So please do that so that you're not completely lost and confused as we move forward in this little mini lecture. That being said, we are going to talk about spermiogenesis and spermatogenesis and possibly semen rather in this little lecture. All right, so we have a couple of different terms that we do need to keep straight. The first one is going to be spermatogenesis. Spermatogenesis is production of sperm. And so what is happening during production of sperm is we are taking large germ cells, which we talked about being located on the edges of those seminiferous tubules, and we are going to remodel them through the process of meiosis into sperm. So our sperm cells are going to be the cells that are used from the male reproductive system, introduced into the female reproductive system, and contribute to production of offspring. A sperm is going to only have 23 chromosomes. We call them haploid cells because they are 1N, which means that they just have one copy of each chromosome. And so part of the process of sperm formation is going from a germ cell, which has 46 chromosomes and is diploid, or 2N, into this haploid 23 chromosome cell. So we do need to reduce the number of chromosomes. And the other important thing, and I mentioned this I think on the very first slide of this lecture, is this idea of shuffling genes to produce genetically unique offspring. So this is sort of the overall goal or series of events that leads to our sperm. Overall, meiosis is going to result in 4 haploid daughter cells, and this is only in the male reproductive system that we see this exact thing happening. I really don't like when it does that. So we start here is our germ cell, so located at the edges of our seminiferous tubules, 2N or 46 chromosomes. It is diploid. We are going to go through 2 rounds of division. In the end, and actually this is showing the egg, but you can pretend that these are sperm. In the end, we're going to end up with 4 daughter cells. The 4 daughter cells are going to be much smaller than our original parent cells, and they are going to be 1N, or they are only going to have 23 chromosomes. We have to do this because if mom and dad are going to contribute equally to an offspring, what we don't want to happen is, so here's generation 1. We have mom with 46 chromosomes, and then we have dad with 46 chromosomes. That's going to give us a generation 2 offspring that has 92 chromosomes. Okay, if they're fertilized by somebody who has 92 chromosomes, then in generation 3, do you see the problem here? We're going to end up with 184. Right? That's an issue. So what we need is we need each generation to have the same number of chromosomes. So rather than do it like that, we're going to make sure that each individual is producing egg or sperm or gamete that has only 23 chromosomes, so just half. So each parent, mom is going to contribute 23 and dad is going to contribute 23. That gives us 46. But then this person, when they make gametes, they're only going to make gametes of 23. Okay? So this way, we can keep the same number of chromosomes generation after generation, and each individual is going to contribute the same number of chromosomes to offspring. All right, so here is probably the slide we're going to spend the most time on because we are going to walk through spermatogenesis. I'm just going to walk through it with words first and then see how that goes, and maybe I'll write things down. But we are going to start with step 1 out here. So step 1 is going to be just our germ cells. Now, germ cells in males are called spermatogonium. So spermatogonia are going to actually be stem cells. Stem cells are dormant before puberty, so they just sort of hang out and they don't do much. They are near the periphery of our seminiferous tubule, so we are inside of a seminiferous tubule now. Here's where we are, so we're on the edge of that. These are going to go through mitosis. So our spermatogonia goes through mitosis, and it's going to form 2 different types of daughter cells. Let me actually go ahead and just write this down. So we're going to start with which is our stem cell. It is going to go through mitosis. This is not meiosis, this is mitosis, and it's going to give us 2 different daughter cells. So one of these is going to be a type A spermatogonium. Oops. And the other one is going to be type B spermatogonium. The type A spermatogonium is going to stay at the wall of the seminiferous tubule and remain a stem cell. Okay, our type B spermatogonium is going to migrate inward. So it's going to migrate towards the lumen. It is going to enlarge into a primary spermatocyte. Okay, so it enlarges into its primary spermatocyte, and at this point, it is going to be isolated by the blood testis barrier. After this point, this primary spermatocyte is going to go through meiosis 1, and it is going to become a secondary spermatocyte. The second, I'm going to come back up over here. So the secondary spermatocyte is going to go through meiosis 2. So that's our second division, and it is going to become a spermatid or spermatid. The spermatid is going to undergo the process of spermiogenesis into a spermatozoon. And at this point, it is going to have a tail, and it loses cytoplasm, which we'll see here in a second. So this is sort of the overall process, and maybe having it written out is going to help us a little bit. So we have division of our stem cells, and we're going to get 2 types of cells. So we get a type A spermatogonium, which is going to be a stem cell. It's going to stay at the wall. It's going to go through mitosis again. The other type is our type B spermatogonium. This spermatogonium is still identical to our original cell, so it still has 46 chromosomes, not an issue. At this point, it is going to start moving inwards towards our lumen. So again, we're in the seminiferous tubules here, so it's moving inward. right around here, it's going to go through meiosis 1. As soon as we go through meiosis 1, we need to make sure that these cells are well tucked behind the blood-testis barrier. So we're actually going to tuck this cell behind the blood-testis barrier before meiosis 1. When meiosis 1 happens, we end up with 2 secondary spermatocytes. So this is a primary spermatocyte, 2 secondary spermatocytes that are now haploid. Each of those are then going to go through another round of division, that's meiosis 2, and they're going to form these spermatids. So these spermatids, they do not look like sperm as we know them, okay, so they look like little cells. The process from being a spermatid to being a sperm or a spermatozoon is going to be called spermiogenesis. Different from spermatogenesis, spermiogenesis is going to be the change from this cell into this cell. Okay, so by the time we have sperm that are sperm-like, they're going to be located at the very edge of the lumen, and then they will eventually be swept down that lumen and out towards those efferent ductals. So here we have shown for us the process of spermiogenesis. So this is where we're going to take those spermatids, which just look like sort of cells, normal cells, and we are going to change them from that into what we would consider to be a mature sperm. Essentially what happens here is we need our sperm to be very small. We need them to contain very little. All they need is our nucleus, mitochondria, and also an acrosome. Those are really the only 3 pieces. We have very little cytoplasm. We have very few organelles. Over time, we're going to go from this big cell that has all sorts of organelles and all sorts of things, and we are going to grow our flagella, so we grow the tail, we shed our excess cytoplasm, we only leave our nucleus, which is going to be in the head, we only leave a bunch of mitochondria, which are going to be part of the mid piece and the tail then, and then our acrosome. The acrosome is going to contain enzymes. And these enzymes are going to be necessary for fertilization. And so this is going to be the process of spermiogenesis. So going from a big sort of blob of a cell to a cell that's very light and small and streamlined and only has the bare minimum that it needs to fertilize. So in the nucleus, it's going to have the chromosomes, those 23 chromosomes to fertilize. The acrosome is going to help penetrate the eggs. The mitochondria are going to provide it with energy so that its flagella can flap and move. Okay, and so here is just sort of our rundown of what the spermatozoon looks like, right? We all know sort of what the shape is. The nucleus just has one set of 23 chromosomes. Our acrosome, like I said, is enzymes. And then the body or the midpiece here is going to have our mitochondria and then our flagella for flapping around. Okay, so I am going to go ahead in this little mini lecture and talk about semen, and then I think the last little mini lecture will be pretty quick because we'll just be going through male sexual response. So we're going to see those 4 steps again when we talk about female reproductive systems, so I probably won't spend a ton of time on them. So we've talked now about pretty much all the pieces of semen. During ejaculation, a male is going to release 2 to 5 milliliters of fluid. Most of that comes from our seminal vesicles. We already looked at these proportions before. The prosthetic fluid, when we look at that, is sort of thin and milky. And these are primarily what it contains. So it's got calcium, citrate, phosphate ions, a clotting enzyme, and then a protein hydrolyzing enzyme. Seminal vesicle fluid is going to be thicker, so more viscous, and also yellowish. It contains fructose, carbohydrates, and then citrate, prostaglandins, and prostaglandin. So let's look at what some of these things have to do with each other. Semen itself is going to be fairly sticky, okay, and so sticky comes from the combination of well, it comes from different molecules that are in either prostatic or seminal vesicle fluid, and sometimes the two have to come together in order for us to get these characteristics. So we get a clotting enzyme. Clotting enzyme is going to come from the prostatic fluid, and it is going to activate the proseminogelin, which is coming from our seminal vesicle fluid. So the two of these are going to be separated in the male reproductive system, but in the female reproductive system, when they're mixed as sperm, or in semen rather, they are going to create this protein. That protein is going to entangle the sperm and also help the semen stick to the walls of the vaginal canal. So it doesn't just slide out, but rather it sticks in there, and it allows time for the sperm to actually move their way from the vaginal canal into the uterus. We also help promote uptake, and it doesn't stay that way forever. So about 30 minutes after semen is deposited, we get another enzyme from the prostatic fluid that actually breaks it down and helps eliminate it. So the other things that we have, we do have a couple of things that help the sperm become motile. So elevated pH and an energy source. So prostaglandins are going to help thin out our cervical mucus in the female reproductive system, and also potentially help with waves of uterine contractions, which are going to help sort of draw the sperm up into the female reproductive system. The pH of the vaginal canal is between 3.5 and 4, so it's going to be very acidic. The prostatic fluid has a buffer in it. It's going to bring that pH up to about 7.5. So again, that's going to help those sperm live. And then also in the seminal vesicle fluid, we have fructose and other sugars. Those are going to be used by the mitochondria of the sperm in order to produce energy and whip that tail around so that they can actually get up into the uterus. So again, a combination of the fluids that are coming from the prostate, from the seminal vesicles, and from the testes all have to work together to make a system in which sperm can find their way into the female reproductive system. All right, and that is it for this. I will come back here in a few minutes and finish up with male sexual response.